Hello, everybody. Thank you for coming to our talk on what feels like the first day of winter. As Julia said, I'm a bioinformatician here at OXCR, and I'll be talking to you today about our work interpreting clinical cancer genomes. There's our next slide. Okay, so today I'll be talking about our motivation for doing this work. Who makes up a relatively new group at OXCR, the Clinical Cancer Genome Interpretation Team. I'll discuss some tools for genome interpretation, walk us through an example case, and we can point to our next direction. So when looking at next generation sequencing, this has allowed us to query genomes at a massive scale, and we're generating larger and larger amounts of data. Where we were once treating patients just based on the tumor or cancer type, we can now create informed decisions based on the mutation profile of each patient, with the ultimate goal being clinical actionability. For example, in a case of lung adenocarcinoma where there have been serial biopsies with tumor profiling, each of these biopsies can be used to inform treatment. So in the first case, there is a host of FDA-approved treatments available to the physician, and in sample 2, the second biopsy, a resistance mutation is observed, and again, based on this genetic profile, there are options available to the physician. Now, it wouldn't be a bioinformatics talk if we didn't have a look at this wonderful slide. And ultimately,
just to say, this is the second time in the past hour and a half that I
it gets
went to another. That's how important.
It's so. I was put onto the slide, and I thought, of course. So we're creating all of the data that's available to us as bioinformaticians, and it's cheaper and cheaper to sequence. However, what they don't tell us is that this might just be how I feel, but it's the cost per analysis could get out of control if we don't look at efficient ways to really analyze all the data that we are generating. So we have to work to efficiently curate clinically meaningful mutations that can be targeted for treatment among the many mutations that are found in the genome, because this is now seemingly our mutation bottleneck. And what we are looking for is driver mutations, which is any alteration previously known to cause cancer, as well as actionable mutations, which may have a clearly defined course of action. So my background here is I started at OXCR as a bioinformatician working in the Translational Genomics Laboratory under Dr. Trevor Pugh. And here we did population or cohort level studies where we worked with different collaborators in the community to do population level studies. There's a bit of a new structure here at OXCR, which many of us are aware of. Trevor is now the director of genomics, and he takes care of Princess Margaret Genomics, the OXCR Genomics Research Platform, and the Translational Genomics Lab. And I guess my group sits somewhere in there. I hope I got it correct. And this is all encompassing genome sequence informatics. So again, what we're hoping to do is once the research team studied cancer from the cohort level, identifying novel prognostic markers, we now aim to take all that we have learned to create a CAP accredited report, which can be signed out by our director, Dr. Trevor Pugh. So this team will review clinically generated reports. We're going to work with clinical geneticists to help them to better understand the data, and we're going to work to continuously identify bottlenecks, so we can try our best to streamline the genomic interpretation process. And I guess I would have clicked on that a bit earlier. Here is, we're aiming to create a clinical report. So this is a grossly oversimplified version of what a pipeline might look like. I actually took out all the tools so that we can focus on, if you've ever heard someone in our group talk before, you might hear about these things we call blue boxes. These are the interpretable components, which we are really focused on analyzing and distilling down as efficiently as we can into a report that will be clinically meaningful for both the patient and the physician. So we're looking at things like mutation burden, different somatic mutations, mutation signatures, fusions, and I'll get more into what some of these mean in a bit later. But on top of the these interpretable components, which are output from the different bioinformatic tools, there's two other things which we need, which I'll be expanding on. We're looking at a reporting software, and we're also looking at a knowledge base, which is kind of a curated collection of all the mutations, as well as different therapies or clinical trials, and also our experience gained from past sequencing. So to do this, we've been looking at the different interpretation practices in the field, and generally what we've reviewed is that there's usually a gene panel of what's been determined to be a clinically relevant set of maybe 100 to 500 genes. There would be a step reviewing patient history, a step looking at biological and technical QC, and I'll expand what I mean by that a bit later, and also a review of the genomic landscape. This could be really any set of oncogenic somatic mutations or resistance markers, whether they're single nucleotide variants or indels, as well as recommendations for treatment and clinical trials. And this is where we've sort of started to grow what we are choosing to do from. And they all tend to have a relatively consistent format. I put up two options here, one from Foundation 1 and one from the Garden 360 group. I think we looked at these last week or last month, the speaker from the AO Monday talk. And these are both gene panels of different sizes, again, from 1 to about 500 genes. There's a consistent format here with the patient and sample information at the beginning, and then it gets into specific mutations which are found in the sample, and it would detail therapeutic implications where appropriate. These commercially available panels, they do an excellent job of solving well-studied cancer types, such as EGFR activating mutations in lung adenocarcinoma, but these are only clinically impactful in a subset of patients, and this is why we are planning to expand this field to include whole genome and whole transcriptome sequencing. One group which we think did do an excellent job is the personalized oncogenomics group at the BC Cancer Agency. They've created an integrated pipeline report, and we've worked with them a lot to inform the direction that our platform wants to go. Again, it takes a similar approach with patient information and going into mutations, and their platform does take it a step further, looking at whole transcriptome, as well as fusions and some expanded information on quality control and cellularity. A lot of this, it wasn't in fact in-house scripts and wasn't terribly portable, so we are working to go our own direction with this. And when I say this would be an example of our new plan. So an example, in addition to the 3 steps which I discussed earlier, we're hoping to expand what's traditionally done in the community with integrated use of copy number variation and expression analysis, as well as looking at structural alterations and fusions, and then ultimately transcriptome analysis, looking at different clinically important pathways, where we can then put this all together into a report which can be passed on to a physician. So why is the current use of a smaller panel? Maybe that's not the best approach. With whole genome sequencing, we can more accurately estimate tumor mutation burden. Obviously, we have the largest sample across the entire genome. We can more accurately determine tumor mutation signatures, which are important in a variety of cancer types. And we can look at identifying novel mutations in oncogenes, which aren't explicitly found in the exons of what's frequently included in a smaller panel, looking at mutations in promoters and regulatory regions. As far as why it's important to include whole transcriptome sequencing, we can characterize novel fusions, we can characterize the immune landscape of a tumor sample, and we can work to identify cancer pathway activity. So what this might look like when we put it all together, we would get a request from a hospital or one of our collaborators. This would undergo a whole genome and whole transcriptome sequencing. The pipelines here at OICR would do a lot of the crunching. And then with a knowledge base, which I'll speak to momentarily, as well as clinical information required for proper interpretation, fed in from a REDCap instance. Our group, as well as some other clinical molecular geneticists, would get together to create a report, which can be signed out by Dr. Pugh and passed on to the physician to ultimately, hopefully, best inform patient care. So this knowledge base that I've been speaking to, I guess it's a topic that we frequently came across when we're kind of looking as to how best do this. Traditionally, we've used databases such as HIVIC or OncoKB, My Cancer Genome. There really is a host of options available out there, but we're looking at ways to best collate this all together, as obviously the scope of looking at clinically important mutations can be extremely large. In addition, we've had a lot of success in the past here at my working group using the vast amounts of genomic data that we've created to create aggregate statistics where we can look at different outliers. We've created blacklists for sequencing artifacts. And that's how to best do this is still an important topic which we are working on. So now I want to demonstrate what this application might look like in a clinical case. And this is a case that may not have been resolved with the traditional approach of looking at a smaller gene panel. So this is a female case, 52 years old with ovarian cancer. And the first step that we would do is review the patient's history. Here we look at important information like clinical history, cancer type, and previous treatment, as this would all help us orient our scope for interpretation. The second step, we would look at biological and technical QC. Here we would look at metrics such as coverage and contamination, and we would flag unexpected mutations, such as unexpected mutations which may identify things like a sample swap or whether or not the mutation is a outlier for a given cancer type. The next step, we would look at the genomic landscape. This is a screenshot from CBio portal where we host a lot of our mutations, a lot of our cases, and make them publicly available to the requesting physician. And in this case, we can see that no oncogenic mutations are observed. So that's why it's important to continue this beyond just looking at somatic mutations and integrate copy number variation and expression analysis. So in this plot down at the bottom, we can see that it's a plot of a copy number track on the bottom and z-score expression on the y-axis, and we can see that there's overexpression of some potentially clinically relevant genes, BREB and LAMP1, and these can help us to drive a hypothesis for further analysis. So based on these findings, we can make a prediction of which cancer pathways are potentially involved. So we can see that BREB and LAMP1 are upregulated, and these are important kinases in the mTOR pathway. And from here, we can then design different experiments to test our hypotheses. So a next step for this might be looking at gene set enrichment analysis to see if the mTOR pathway could in fact be involved. So gene set enrichment analysis, it looks for signatures of whether or not certain pathways may be significantly activated or deactivated in a given sample. And this in this case, we can see that there is good evidence that mTOR signaling is involved, and as a result, we could make a suggestion to a clinician that this mTOR-based upregulation could be a significant player in this cancer type, and then obviously this would be up to the physician as to how to best use this information. So now I just want to quickly discuss more about these newer tools and how we want to incorporate this approach into our work. So the first would be looking at, as I mentioned, integrated copy number variation and expression analysis. We're particularly interested in copy number changes, which have an accompanied change in expression. And we're also interested in looking at truncating mutations where there is a loss of expression. And again, it's still quite a manual process, but these can be clues to help drive our hypotheses for further pathway analysis. This is a plot from a tool called Robotic, which we are working on. And it was worked on a lot by John, who's in our group, as well as our Waterloo Co-op student, Syed. And it plots copy number results against the z-score. So the next thing we're interested in applying is looking at and characterizing structural alterations and fusions. For this, we're using a tool called MAVIS, which can connect structural variant callers from our whole genome sequencing workflow with fusion candidates from our whole transcriptome workflow. And in this way, we can validate novel fusions without too much work for the downstream validation, as it can merge the annotations and we can get evidence from both pipelines. We're also interested in integrating transcriptome analysis, and we're looking at a variety of ways to best implement this. One such way we're looking at implementing transcriptome analysis is through a RNA-seq tumor classifier. Was primarily worked on by Alberto, who works in our research group. This may be used for quality control to ensure that it's the cancer type that we are interested in investigating, because misclassification could potentially change our interpretation dramatically. And it's also a functional check to see if the data is well formed for our further downstream analysis. And we're also interested in looking at a variety of other expression outliers, pathway analysis, as I mentioned earlier, as well as immune interference. And as we aim to continually develop and evaluate different tools, we hope that we can incorporate more of these practices into our CAP validated report and workflow, such as making comments on the immune profile and the different cancer pathways which are potentially involved for a given tumor sample. So with that, I'd like to thank everybody from the Genome team. It's now a much larger team. I'm still getting to know everybody, and especially the other two members of this brand new team, which is Alberto and Jonathan and myself. So thank you very much for listening to me this morning.